Welcome everyone to the second day of our fifth annual Children's National NIAID Symposium focused on COVID-19. And this year, our topic of interest, autoinflammatory and neuroimmune disorders. This is the second day, as I mentioned, and this day will pivot from a fantastic set of discussions yesterday regarding COVID-19 to an equally exciting set of discussions about autoinflammatory and neuroimmune disorders. As we discussed yesterday, this is now an exclusively virtual meeting, and so we've interpolated several abstracts that were submitted into the agenda for today. And so at the end of each of the sessions, we will have abstract presentations. Yesterday were terrific presentations, including one by an intrepid high school student that I thought was just great. For those of you who have questions and answers, please use the question and answer function on Zoom, and we will be sure to try to call out your questions in the question and answer periods. And if not, then I'd ask all the speakers to take a look and answer the questions that are posed. Speaking of questions, our first speaker is our keynote speaker, and Dr. Kastner has graciously agreed to take questions at the end of his presentation. So let me go on and introduce our keynote speaker, Dr. Dan Kastner, who will speak to us about the inevitability of autoinflammatory disease. And so it's a personal pleasure after years of knowing Dr. Kastner to introduce him. He is the scientific director of the Division of Intramural Research at the National Human Genome Research Institute. He obtained his bachelor's degree summa cum laude in philosophy from Princeton, and then his MD PhD from the Baylor College of Medicine. He then went on to complete an internal medicine residency and chief residency at Baylor, and then scooted off to the East Coast and joined the NIH. Where he has developed a really illustrative research career. He's focused on using genetic and genomic strategies to understand inherited disorders of inflammation, often stimulated by patients with relatively rare disorders that are seen at the NIH Clinical Center. So he is really a true physician scientist. His work has provided detailed molecular explanations for many of these illnesses, laid the foundational predicate for highly effective targeted therapies, and informed our understanding of more common immunologic illnesses. Dr. Kastner's group was the first to propose the now widely accepted concept of autoinflammatory disease to characterize and describe disorders of the evolutionarily ancient innate branch of the human immune system. For his many research accomplishments, Dr. Kastner has won a number of awards and honors, including election to the National Academy of Sciences and the National Academy of Medicine, recognition as Federal Employee of the Year, which is really a coup, and the Ross Prize in Molecular Medicine, most recently in 2019. And so without further ado, I'll turn the microphone over to Dr. Kastner. Dan, we're delighted to have you.
Well, Lisa, thank you so much for that kind introduction. And I must say, it is just a joy to be with you guys this afternoon and have an opportunity to talk about these really interesting and I think informative disorders. So for anyone in the audience who doesn't know about the autoinflammatory diseases, perhaps we should start there just by giving a brief definition. These are disorders that were initially recognized as being conditions with seemingly unprovoked inflammation without the high titer autoantibodies or antigen-specific T cells that one would see in the classic autoimmune diseases like lupus, for example. As time has gone on, it's become clear that the monogenic autoinflammatory diseases, in fact, are caused in many cases by mutations in genes that encode either structural proteins or regulatory proteins involved in the innate immune system, as Lisa referred to. Whereas the autoimmune diseases, the monogenic autoimmune diseases, are disorders in which there are mutations in structural or regulatory proteins involved in the adaptive immune system. Now, we're going to be talking this afternoon about the inevitability of autoinflammatory disease, and I'll try to convince you that at least at some level, the autoinflammatory diseases are inevitable because of the fallibility of DNA replication, leading to inborn errors of innate immunity, because of selective pressure, sometimes exerted by pathogenic organisms that select for variants in the innate immune system that will lead to increased resistance to the pathogen, but may also lead to autoinflammation. And then, of course, there's also the possibility of somatic mutation. So with that having been said, let us move on. Let me see if I can get this to, yes, to that replicate first slide, that requisite first slide. I have nothing to disclose. And then moving on to the first section of the discussion, we'll talk a little bit about inborn errors of innate immunity, which in some cases can appear to be inborn errors of cell biology. The first example of this that we'll talk about is just briefly a paper that we published in Nature about a year and a half ago on a new autoinflammatory disease that we call CRIA syndrome. CRIA standing for cleavage-resistant RIP kinase-induced autoinflammatory disease. The story for this actually goes back a couple of decades, and we've seen over that period of time 3 families in which the common feature has been recurrent fever with painful diffuse lymphadenopathy and organomegaly. And that's simply illustrated in these images on the lower left. These 3 families have in common that they have mutations in RIP kinase, which is an important signaling molecule in the immune system. And in each case, for each of the families, the mutation is at position 324, which happens to be the site at which RIP kinase should be cleaved by caspase 8. And in the first family, there's a mutation of asparagine for aspartate at position 324. In the second family, a substitution of histidine for aspartate at position 324. And in the third family, the substitution of tyrosine for aspartate at position 324. This is a highly conserved residue across evolution. And if one looks in transfection systems, one can see that one gets cleavage of the transfected RIP kinase where one should get cleavage, shown here with the wild type, but not for a mutant that is uncleavable, deliberately uncleavable. And then the transfected variants that are seen in the patients are also uncleavable. This leads to hypercytokinemia in these patients, high levels of TNF, IL-6, and IL-1 beta. It leads to an inflammatory gene expression profile, and at least some of these patients are responsive to treatment with tocilizumab. Now, in collaboration with a group in Australia, we made knock-in mice for these mutations. And in fact, the knock-in mice for uncleavable RIP kinase also have very high levels of TNF, IL-6, and IL-1 beta. But that affords us the possibility then of breeding these mice with knockouts for various molecules in the immune system so that we can dissect mechanism. And when one breeds these uncleavable RIP kinase knock-in mice with knockouts for RIP kinase 3 and caspase 8, which basically blocks the apoptosis and necroptosis cell death pathways, then you get a reduction in the amount of TNF in the blood, and you also get a total abrogation of the amount of IL-1 beta that's produced. And so this is telling us that basically RIPK1 cleavage, which had been a mystery up until the identification of these patients, is actually a molecular switch between cell survival and the apoptosis necroptosis induced inflammation. Now, lest you think that this is just a curiosity that happens just in this one particular situation. In fact, here's another paper that was just published last year in Cell by Dusan Bagunovich's group, looking at mutations in TBK1, another important molecule in the immune system. And many of you may know that TBK1 is involved in generating type 1 interferon responses to viruses. But a second action of TBK1 is actually regulating RIPK1. And when you have loss of function mutations in TBK1, that actually unleashes RIPK1 to go down the apoptosis, necroptosis dependent cell death pathways. And in fact, one develops then inflammation based on that. And in fact, it's a TNF dependent process. So patients that have those mutations do respond to TNF inhibition. What's more, here's a third disease, and this is unpublished work on a new disease that's caused by mutations in a molecule called Sharpen. And Sharpen is important in the LUBAC complex, the linear ubiquitin assembly complex. And because there's a deficiency in Sharpen, we call it Sharpenia. We first encountered this in a young man from southern India, from Bangalore, who's actually the son of an uncle-niece marriage. He had a blood sample sent to us. He was in his teens at the time we got the blood sample because he had unexplained recurrent fevers, he had parotitis, he had arthritis, and he had a form of colitis. When we did exome sequencing on him, we found that in fact he has a frame shift mutation in Sharpen, a loss of function mutation in Sharpen. And so that would be predicted then to cause problems with linear ubiquitination. And in fact, there were mice that had already been recognized just by chance at, I believe it was Jackson Labs, that had a mutation in Sharpen, that had an inflammatory phenotype that actually was dependent upon TNF. That is to say, breeding those mice with TNF knockouts led to abrogation of the inflammation. So we thought that maybe we could treat this young man with TNF inhibitors. We brought him to the NIH to examine him to see whether or not, in fact, he had the inflammatory process that we had heard about. Sure enough, on these scans, he had arthritis of his ankle joint. He had arthritis of the atlantoaxial joint, which of course could be fatal. And he had colitis shown here on this colonic biopsy. What's more, we found that he did not have any evidence of opportunistic infection, which of course would have been a concern if we were to start a TNF inhibitor on him and he had an opportunistic infection. So in any case, gradually, we did put him on a TNF inhibitor, but before doing that, we did confirm that he had reduced canonical NF-kappa B signaling, that is to say, cell survival NF-kappa B signaling in his leukocytes, but had increased cell death, Sharpen-induced, TNF-induced cell death in the individual with a Sharpen mutation. So again, it's the same idea that there's a shunting of inflammatory responses towards a cell death dependent pathway, which can lead to autoinflammation. So we started him on a TNF inhibitor, at least initially beginning with etanercept and then eventually switching over to adalimumab. You can see that his arthritis resolved, that his colitis resolved. We got him back on the growth curve, both in terms of height and in terms of weight, and his bone mineral density also improved. When he first came to us at the NIH, he was in a wheelchair. And over the course of a year that he was on TNF inhibition, you can see he's standing here in clinic. And in fact, we put him through his paces, and you can see that, in fact, he was able to dance. So it really a very, very gratifying response, and another vindication of molecular medicine, that in fact, here's a TNF-dependent inflammatory process. One last inborn error of innate immunity. This is also unpublished work that has to do with a new disease. Another group has published this disease, but we're now investigating it further. A disorder called Rosa Syndrome. And Rosa Syndrome stands for retinal dystrophy, optic nerve edema, splenomegaly, anhidrosis, and migraine headache. And it's caused by mutations in a gene called ALPK1, which encodes a protein that actually is a sensor for 7-carbon sugars, an innate immune sensor for 7-carbon sugars. Why is that important to have a sensor for 7-carbon sugars? Well, bacteria can make 7-carbon sugars. We can't make 7-carbon sugars. So to have an innate immune sensor for 7-carbon sugars allows cells of the immune system and other cells in the body to detect bacterial infection. And actually, this ALPK1 protein is a part of a family of kinases, which is relatively unusual. The autoinflammation that these patients can develop is quite interesting and dramatic. Here is the optic disc edema. These patients can have calcifications of the basal ganglia. They can have a polyarticular arthritis, massive splenomegaly. And basically, this is caused by gain of function mutations in this ALPK1 protein, which then leads to activation of the canonical NF-kappa B pathway and activation of inflammatory pathways as well, other inflammatory pathways. We're still working on this in terms of developing a targeted therapy. We know that various cytokine inhibitors can have an effect on the inflammatory manifestations, but the vision loss is still somewhat of a mystery to us. We have some promising data with regard to the use of the IL-6 blocker tocilizumab, but it's still early days with regard to that. At this point, though, I'd like to move on to 3 other little vignettes of things that we're doing in the lab that further speak to this question of inevitability of autoinflammation. And as I think made the case that there's plenty of inborn errors of innate immunity out there that support the idea of inevitability of autoinflammation. But now let's talk about natural selection a little bit. And we were asking the question, why are there such high carrier frequencies for familial Mediterranean fever, which is, of course, one of the prototypic autoinflammatory diseases in multiple Middle Eastern populations? And you can see on this table here that, in fact, the total carrier frequency for FMF mutations, severe FMF mutations, in 4 different Middle Eastern populations, Turks, Armenians, Arabs, and non-Ashkenazi Jews, adds up to between 10 and 12%. To put that in some sort of perspective, the carrier frequency for cystic fibrosis, the most common lethal recessive disorder in North America, is about 4%. The carrier frequency for the sickle cell trait in the African American population is about 8% or so. So 10 to 12% is a whopping percentage. It does make you think that maybe there was something selecting for it. But in order really to establish a case for selection, one has to do some serious population genetics. And so the way of doing that is to look at the size, the extent of the DNA haplotype that's associated either with the mutant or the wild type form of the gene in one of these Middle Eastern populations. And we're looking at the width of the peak here as a descriptor of the size of the associated haplotype. So blue represents the mutant haplotype, the M694V mutation in this particular case. And you can see that the width of this is quite extensive versus the width of this peak, which is associated with the wild type. That's the kind of thing that you see with selection. What's more, one can use this information to actually calculate a coefficient of selection. And the coefficient of selection determined here on this graph for this particular mutation in the Turkish population is 0.077. To put that in some perspective, the selection coefficient for lactase persistence, that is to say, the ability to digest cow's milk as an adult, something that we know has been selected for in the Northern European population, is 0.056. So 0.077 is a high selection coefficient. And one can even then estimate the time at which these FMF mutations arose, and actually using 3 different methods, we come up with an estimate of between 2,000 and 5,000 years ago. Well, there's a lot of molecular biology and bacteriology and immunology that we won't have time to talk about, but basically the idea for why these mutations have been selected for, thinking about the organisms that might do it, certainly one that was an attractive hypothesis was Yersinia pestis, the agent of the bubonic plague. And it turns out that Yersinia pestis makes some toxins, YAP-E and YAP-T, that actually activate pyrin, the FMF protein, and induce pyrin to produce interleukin-1 as a defense against the plague. But actually, pathogenic Yersinia pestis also has a toxin called YAP-M, which induces the phosphorylation of pyrin, which actually then blocks the production of interleukin-1 beta. For patients that have FMF, the mutations that are associated with FMF in pyrin actually render pyrin resistant to YAP-M, so that mutant pyrin that's associated with familial Mediterranean fever actually can lead to the production of IL-1 in response to Yersinia pestis, to the plague. But that's only in less than 1% of the population. But it actually turns out that carriers who are heterozygous for the mutation are also resistant to that effect. And that's about 10% of the population. So you have this Faestian bargain, if you will, in which 1% of the population has a mutation that can lead to a disease that can be fatal, familial Mediterranean fever, if untreated, at least in some parts of the world, can lead to amyloidosis, which is fatal. But in exchange for that, you have the carriers who represent a much higher percentage of the population for recessively inherited disease, who are also protected. So basically, the Faestian bargain is that 1% of the population gets a disease which can be fatal, but that protects 10% of the population from at least some aspects of that disease. So there are ways then that pathogens can select for variants in innate immune molecules that can lead to autoinflammation. And we can actually simulate the carrier frequencies for FMF based on this information, on the carrier frequencies, on the selection coefficient, on the history, and actually come up with estimates of the carrier frequency, which are very close to what we actually observe. A second riddle of the Sphinx is, how do old people develop autoinflammatory disease? This is the work of a former fellow in our group, David Beck. And David was interested in taking sort of a different approach to disease gene discovery. Usually what we do is a phenotype first approach, in which we have a whole bunch of individuals that may have fevers of various sorts or whatever, and we'll pick out a subgroup of them that have some particular phenotype that we're interested in, and then find susceptibility genes that lead to that phenotype. That's the phenotype first approach. David decided to turn things on their head, and instead of starting with a phenotype, he started with a genotype, and then tried to identify individuals who have mutations in that gene. So that's the genotype first approach. And he actually had a set of 2,500 exomes from our lab and from the Undiagnosed Diseases program that he interrogated against over 800 genes that are involved in ubiquitination, a process that he thought might be associated with a new autoinflammatory disease. And what he found was that, in fact, there is a gene, UBA1, which is a master regulator of ubiquitination, somatic mutations of which cause an autoinflammatory disease. And just on this slide, we can see that in the peripheral blood, there is both the mutant and the wild type form of this UBA1 gene. But if one looks in the myeloid cells, there's just the mutant form, and in lymphocytes, there's just the wild type form. So it is actually an example of somatic mutation, where only a subset of the cells have the mutation. And knowing that, David was then able, through the rheumatology clinics of the NIH, to identify some 25 patients relatively quickly who had mutations in this gene, who had just a variety of different adult onset inflammatory diseases, including relapsing polychondritis, Sweet syndrome, myelodysplastic syndrome, MGUS, polyarteritis nodosa, and giant cell arteritis. But because they all had mutations in the same gene, he gave this condition the name VEXAS. And VEXAS stands for vacuoles, E1 ubiquitin activating enzyme, X-linked autoinflammatory somatic syndrome. And these patients have a number of clinical manifestations. They are, because this is an X chromosome encoded gene, they are men, nearly all of them, and usually they present after the age of 40. So this is not for pediatrics necessarily, but the principle still applies to pediatrics. And you can see that these patients can have pulmonary infiltrates. They can have neutrophilic infiltrates in the alveolar spaces. They can have medium vessel vasculitis. They can have polychondritis. They can have these funny looking vacuoles in the precursor cells in the bone marrow. But the big picture is that basically this is a paradigm shift in gene discovery, using a genotype first rather than a phenotype first approach. Adults may develop autoinflammatory disease the same way that they develop cancers, by somatic mutations. And throw out the textbooks, because in fact, we have now an example of a genetic mutation that defines the disease that cuts across the clinical diagnoses that we had used before. And this may turn out to be something that we'll see more and more in adult onset autoinflammatory or even autoimmune diseases. Finally, the third riddle of the Sphinx. What is the genetic mechanism of common autoinflammatory conditions? And I've been interested in this. I was having dinner with a friend in Istanbul back 15 years ago or so, when we were thinking about how to approach this question. And we decided that Behçet's disease would be an interesting way of looking at it, because we know that it's a genetically complex disorder. Of course, patients who have Behçet's have the triad of painful oral ulcers, ocular inflammation, and genital inflammation. They can also have the pathergy phenomenon, where they get a pustule when you prick the skin with a sterile needle. Of course, Behçet's disease, idiopathic Behçet's disease, is distributed along Marco Polo's Silk Route, going from the Mediterranean basin through Asia to East Asia, Korea, and Japan. So we know actually that there are multiple ways that you can get Behçet's disease. There's a monogenic form of it, haploinsufficiency of A20, that we won't get into. But more often, Behçet's disease is associated with common variants in a number of genes that are involved in innate or sometimes adaptive immunity. And it's almost as if it's a Chinese menu kind of situation, in which you can have various combinations of variants in those genes that will lead to Behçet's disease. Well, having found that, we were astonished shortly after assembling this list of variants, common variants that are associated with Behçet's disease, to learn that the direct-to-consumer DNA testing company, 23andMe, had done a study of the genetics of canker sores, just common canker sores. And they had found that actually a number of the genes, that variants of which, common variants of which, predispose to canker sores, are the very same genes that we had found associated with Behçet's disease. Of course, there are some genes that are associated with Behçet's that are not associated with canker sores, but there is this common core that is associated. And then, what's more, Kalpana, a fellow in my lab, looking at a disorder called FAPA syndrome, which many of you may know about, the syndrome of periodic fever with aphthous stomatitis, pharyngitis, and cervical adenitis, also has variants, these same variants in 4 different genes that are associated both with canker sores and Behçet's disease, the P35 subunit of IL-12, STAT4, IL-10, and CCR1, CCR3. So this leads to the thought that perhaps there is a continuum, a spectrum of disorders. The Behçet spectrum disorders is what Kalpana calls it, ranging from, on the mild end, recurrent aphthous ulcers, to on the more severe end, Behçet's disease. So the take-home messages from this talk. Number 1, the autoinflammatory diseases are disorders of innate immunity. Number 2, monogenic autoinflammatory diseases have taught us a great deal about how innate immunity works. Number 3, in some cases, targeted therapies have been transformative while validating our understanding of the underlying disease. Number 4, pathogens sometimes select for common genetic variants that protect against the pathogen, but predispose to autoinflammation. Number 5, somatic mutations account for some adult onset autoinflammatory diseases. And finally, cassettes of relatively frequent genetic variants confer risk of common autoinflammatory conditions. At this point, I will simply show the pictures, but for lack of time, won't say them by name, all of the people that have contributed so enormously to this work. This is in pre-pandemic times, just showing that this is a collaboration between people in the lab and people in the clinic. A number of collaborators contributed to this, and this is just the clinical center of the NIH where a lot of this work was done. And with that, I will call it to a close and turn things back over to Lisa, who will pepper me with questions, I am sure.
Well, not exactly pepper, but I will congratulate you on an absolutely elegant talk that really shows the power of molecular medicine. That you actually answered the one question that folks submitted, and I think it's because your talk was just so clear, Dan. But I wanted to follow up on one of your points about the genotype to patients. And I wonder about, with these large resources in the UK, with all of us in the United States, where we're going to have huge number of individuals who contribute some of their clinical information, but their DNA. If you're, because I'm sure you're thinking about this, thinking about strategies where we can ask the questions like you asked for the cohort of patients at the NIH, or extending that all the way to direct-to-consumer genetic operations like 23andMe, asking the question about canker sores rather, as a sort of a prototype of this continuum of disorders. So how would sort of the bioinformatically savvy, probably more junior folks in this audience be thinking about mining those big data sets to do the kinds of things that you've done?
Well, that's a fantastic question, Lisa, and certainly it is something that's very much on our minds. And so the All of Us initiative that you were alluding to actually at this point has collected DNA samples and has deposited sequences in a database for some 200,000 individuals, and they're going to be going up to 1 million individuals. And investigators who are at institutions that have signed various assurances with the All of Us program, maintaining confidentiality and that sort of thing, then can get access to that database and do the very thing that you are suggesting, that if you're interested in some particular gene or set of genes that you think may have a particular phenotype associated with it, or maybe you don't know exactly what phenotype would be associated with variants in that gene, you can petition to have access to the sequence data and then interrogate and find out all of the people in that database who have variants in that gene. And of course, there's rich phenotypic information that's also associated with those individuals. And so one can begin to say, okay, well, I'm interested in such and such gene that has something to do with inflammation. And by golly, patients who have variants in that gene have a certain kind of skin rash or arthritis or, whatever. So it is a very, very rich and potent tool, engine for the possible discovery of new diseases.
Great.